Welcome, members. Thank you so much for joining us today. We are going to go ahead and get started. We have a big group of us joining, but we will be meeting with Elora López-Nandam, an Academy postdoctoral researcher in the Coral Regeneration Lab. Elora will be sharing with us more about the Academy's Coral Regeneration Lab, what we can learn from coral genomics, and some exciting details about this year's spawn. We'll have plenty of time for Q&A at the end, but please feel free to add any questions in the chat box. There are also some instructions in the video description on how to join the YouTube chat. Well, without further ado, let's bring Elora on to start her talk. Hi, Elora.
Hi, Jess.
Awesome. Thanks for joining us today. So excited to learn more about our corals. And I'm going to let you take it away.
Thank you so much. This is really exciting. And thank you everyone who's tuning in online to learn about coral spawning, genomics, and how that all leads, we hope, to Hope for Reefs in the Coral Regeneration Lab at Cal Academy. So today I'm going to tell you about what is actually newly branded as the Coral Regeneration Lab. If you've been following our work before, you may remember that we were called the Coral Spawning Lab. We just recently rebranded because we really want to emphasize that while the spawning is really exciting, there's a much broader goal here, right? Of which spawning is just one part that happens once a year. But the science and the goals of coral regeneration are 365, 24/7 goal for us. So we have renamed ourselves to the Coral Regeneration Lab. So today I want to tell you all about first of all, what coral reefs are, why we care about them, and then more specifically, a lot of the work and a lot of the recent work that's been happening here at Cal Academy. So I'm going to start really broadly by asking the question of what is a coral reef? And some of you may know the answer to this already, and for those of you, just sit back and enjoy for a few minutes before we really get into the nitty-gritty. But for those of you who maybe haven't really thought about corals before, I show you this photo that's been taken in space and that shows us an ecosystem from miles above the Earth. So we can see that this is an incredibly large ecosystem. And if we're actually going to zoom in and go under the surface, we start to see what that ecosystem is made up of. So although you see all these pretty fish in the front and the top of this photo here, the thing that I really want you to focus on are the bottom of this photo. So at the bottom of this photo, we see mainly two different species of stony corals. We see these kind of plating structures here, and then some mounding corals over here. And we see that there are these really interesting 3D shapes that provide a lot of habitat and shelter for all sorts of different animals, including fish and snails and lobsters and all sorts of other creatures. But that doesn't really tell us yet what are these things that we call corals. So they kind of look like rocks, they kind of look like plants, but they're actually neither of those things. They're actually animals. So corals are animals that are part of this ancient phylum called Cnidaria. Cnidarians include sea anemones, jellyfish, and then both soft and stony corals. And the ones that I'm going to focus on today are these stony corals. The reason that we call them stony corals is because they create these calcium carbonate skeletons. So that 3D structure that's creating the ecosystem is actually this very hard skeleton on which the very thin living tissue of the animal actually resides. All right, so here we are looking at one coral colony that I was measuring a couple of years ago in American Samoa. So the next question is, why are we calling this a coral colony? So if we zoom into this colony a little bit, you start to see that it's actually made up of all of these little modular components. So what you're looking at here, these little cups, are parts of the skeleton. And then if we're zooming in a little bit more, you see that within each of those cups, there's these little tentacles sticking out. And if we zoom in just a little bit more, we can actually zoom in on a single polyp. So this translucent, kind of soft, colorless structure is the living tissue of the coral. It's called a polyp, and it is in and of itself a multicellular animal. So it has its own tentacles, its own mouth, its own gut. And the way that a colony grows is it starts as a single polyp, and then one polyp asexually reproduces, so it divides into two polyps. And so a large coral colony might be made up of hundreds of thousands to millions to tens of millions of polyps that have all been asexually dividing. So they're all actually clones of each other. So the way I think of a colony sometimes is actually that the skeleton itself is almost like the apartment building, and then all of the little polyps living within the skeleton are the inhabitants of that building. Except, of course, in this case, the inhabitants of the building are all genetically identical. So they're all clones of each other. So that's interesting. The other interesting to note here is that there are a few pigmented cells in this polyp. And these kind of greenish brown cells that you're looking at here are actually not the animal cells, they're algal cells. So coral polyps live in symbiosis with these algal cells that take energy from the sun and convert it into food for the coral. That's a process called photosynthesis. And so this relationship we'll talk about later, why it's so important. But for now, you've learned a little bit about what a coral is. Now we need to talk about why we should care about coral reefs. So one of the big reasons why we care about coral reefs is that they are incredible harbors of biodiversity. So this was a study that was done about 20 years ago now, in which they surveyed coral species diversity across the world. So this second panel here is coral species diversity, with hotter, like bright red being there's lots of different species found in that area, and cooler colors meaning there's fewer species of corals found in that area. And what's been seen is that there's this very tight correlation between coral species diversity and fish species diversity, snail species diversity, lobster species diversity. And a big reason for that is because corals are so good at creating habitat. So if you have lots of different types of corals, then you have lots of different types of spaces for speciation of other different types of animals. So when people talk about corals as being the rainforest of the ocean, this is a big reason why, because although corals only cover about 1% of the ocean surface, over 25% of fish species in the ocean live at some point on a coral reef in their life. And so these are incredibly important ecosystems for biodiversity. From that, we also realized that corals, therefore, are incredibly important for fisheries. So anywhere from really small scale subsistence fisheries where people are fishing, to catch the food that will feed their family that night, all the way up to, industrial fishing, commercial fleets that are going to be, shipping fish across the world really depend on healthy coral reefs. Corals are also really important for coastal protection, and this is something that the US military and a bunch of different other, very infrastructure-minded organizations are starting to really think about. So if you've ever stood on a beach that has a reef, you might have noticed that the waves don't really hit the beach itself. The waves actually break where the reef crest is, which is often, pretty far away from the beach. And so when you lose coral reef, if you lose that 3D structure, then damage due to coastal flooding or erosion or storm damage can really be heightened. And so one reason we really need to think about coral reefs is to mitigate these types of damages. Also, coral reefs are inherently beautiful and people travel all over the world to see them. And so they are critically important for a lot of economies that are based around recreation and tourism that are associated with coral reefs. But beyond that, coral reefs have cultural significance. So there are all sorts of communities around the world that recognize reefs as these incredible systems culturally. This is a line, one of the first lines from the Kumulipo, which is a Hawaiian origin chant. And the line goes, "Born was the coral polyp, born was the coral came forth." And this line emphasizes the coral polyp is actually the first animal to appear on Earth, which, when you think about it, Cnidarians are actually incredibly ancient animals. And so there is this recognition in a lot of cultures as these being incredibly important, ancestral animals. One, I think, often overlooked aspect of why we should care about corals is because they might serve as really great models for genome integrity and cancer research. So this photo that you're looking at here is of a giant coral colony. You can see the person next to it looks very small. So this colony is called Big Mama, and it lives in American Samoa, and it's been estimated to be over 530 years old. So usually when we think about really old, really large animals, we have to think about, cancers and things like that. And yet corals don't seem to get cancers or show aging in the ways we usually think about for animals. And so I've really championed corals as something that we should really be studying, how they accumulate mutations and how they protect themselves in some cases from mutations, so that we can learn about all sorts of different biomedical and developmental biology as well. All right, so hopefully at this point, A, you know what a coral is, and B, you have a sense of why we should care about them. So now we can start talking about what's happening to coral reefs right now. So one big issue is all sorts of different local stressors. So things that can happen, on a regional level. So coastal development, overfishing, runoff from agriculture. I'll also add to this list diseases, particularly if you're thinking about the Caribbean, stony coral tissue loss disease. But then there's also, global stressors that's affecting all coral reefs. So global warming, creating ocean acidification and bleaching. And the thing I really want you to come away from this talk knowing about is coral bleaching. So you may have heard about bleaching in the news in the last 5, at this point, maybe almost 10 years, particularly on the Great Barrier Reef or other really iconic reefs around the world. And bleaching is actually, although it happens on a very large scale, which can be very scary, it actually starts on a very microscopic scale. And so now we return again to this photo of a single coral polyp. As I mentioned before, coral lives in a symbiosis with algae. But for reasons that we're still trying to work out and people are actively studying, so we don't quite understand the mechanism yet, when ocean temperatures rise past a point where the coral is used to or comfortable with, the corals appear to expel their algae. So they actually get rid of them. So the reason that we call it coral bleaching is because they lose those pigmented algal cells. And so the animal now is just, it's white color. So they turn white. This is really a huge problem because corals obtain up to 90% of their food from the algae photosynthesizing. So a bleached coral is essentially a starving coral. And if they don't get their algae back, then they effectively starve to death. So this is an example of what that can look like. This was what used to be a healthy reef in American Samoa in December of 2014. So you see a bunch of the corals, they're kind of this nice yellow, brown, pigmented color. This is what it looked like in February of 2015, just 2 months later. Totally bleached, totally white, loss of algae. And then by August of 2015, this entire reef was dead. So what you're looking at here is a dead reef, and eventually, because the animals themselves have died, that 3D structure will start to crumble, and an ecosystem is essentially lost. So how are scientists trying to protect corals in the face of all of these local and global stressors? And, particularly the major one of global warming. So I would say that the most important factor in how corals will fare in the next century is how much we decrease greenhouse gases by and how soon. But we also know that even if emissions levels dropped tomorrow, ocean temperatures would still be on the rise. Like we are on that course. And so the hope is that restoration efforts may be able to bolster or help coral populations to adapt to new conditions. And so there are two different ways in which people are really trying to do this right now. One is restoration via fragmentation. So if anyone in the audience is a gardener and has worked with, succulents or cacti or anything like that, you may know that you're able to break off, a branch or divide it into a couple of different pieces and reprod it, and that plant will keep growing. The same process is true of corals as well. Since, they're asexual clones, you can break off a couple of branches and have them start growing elsewhere. So that's fine, and people have started to do some restoration that way. But the problem is that there's really no genetic diversity then. And so if a stressor comes by, like a disease or higher temperatures or something like that, if the particular, clonal genotype that's covering the entire reef isn't suited to be able to survive that stressor, then the entire reef can get wiped out very quickly. So the alternative that we at Cal Academy are really excited about is instead taking advantage of coral sexual reproduction. So for the animals that we work with, they're called spawners, and that's because they release their gametes, so eggs and sperm. And yes, in the animals that we're working with, the same animal produces both eggs and sperm. And those eggs and sperm meet each other in the water column. So they fertilize, they become an embryo, and then they become a little swimming larva. And I'll show you videos of all of this if it seems really strange right now. After a few days to maybe about a week, the larva hopefully finds a nice place to settle, and it settles down on the ocean floor and starts growing its first polyp. Eventually, those polyps divide, become a coral colony. So the great thing about this is that it's actually creating genetic diversity the way sexual reproduction does in other animals. The other thing that's really interesting and very strange about corals is that they do their sexual reproduction only once a year, and it's entirely synced with lunar cycles. So which which full moon the coral is associated with varies depending on the species and depending on where you are in the world. But a lot of them tend to only spawn once a year, and it's fairly predictable which full moon in the year they will spawn near. So for example, there's a species in Hawaii where it's known that there's a particular full moon, I believe for this one, it's in the spring, where the corals will spawn within 1 to 3 days of that particular full moon. And if we stop to think about this for a second, it actually makes a lot of sense that if you're an animal who wants to reproduce, but you're stuck to the ocean floor, you can't move to find a mate, it's totally advantageous for you all to release your gametes at the exact same time. So same day, same time, because that maximizes your eggs and sperm chances of actually finding a mate in the gigantic ocean. If you go off at a different time and no one else is releasing, you're probably not going to find a partner. So this gets even more interesting because it's not just the lunar cycle that the corals are synced with then, it's also the circadian cycle. So in addition to being fairly predictable in which full moon they spawn near, we can generally start to predict how long after sunset the corals of a particular species will spawn as well. So they're dialed in not just to the day, but often to the minute, so that all of the animals of the same species on the same reef truly are releasing their gametes within a few minutes of each other. It's pretty remarkable. So how can we start to take all of this information and harness it into a regeneration plan for corals? This is where the Coral Regeneration Lab at Cal Academy really comes in. So our lab was started by my boss, Rebecca Albright. And a few years ago, she and a bunch of other really incredible folks at Cal Academy built this lab space. And so what you're looking at is a set of 6 different tanks that are all being controlled. The temperature is being controlled. The lights that act as the sunlight are being controlled. There's a separate set of lights that act as the moonlights that are being controlled, so that we can accurately mimic all of the different conditions, the lighting, the temperature, all of that, that corals would experience on a reef throughout the year. And by doing all of that, we can actually get the corals to spawn in the lab at the same time that they would be in the wild. So I'm going to show you a brief video about kind of the overview of how all of that works. So this video was created in 2019 around one of the first really successful spawns that happened at Cal Academy. So first, you're seeing the building of the system. So this is the lab itself getting built. And now you're seeing where some of our first corals were sourced in Palau. For those who don't know, Palau is an island nation. It is located in the Central Pacific. And so a bunch of Acropora hyacinthus colonies were taken. So not the entire colony, just a section of it, were taken off of the reef and shipped to San Francisco. And incredibly, after a very long transit, they're still happy and healthy and ready to start spawning. So what you're looking at right now is, in fact, a coral spawn. So these are gamete bundles, so packets of eggs and sperm that are being released into the water in our aquarium system. And people often ask me if these videos are slowed down. They're actually not. So what you're watching is what it looks like in real time. It's kind of this slow release of those bundles. So once the bundles are released, we can collect them and we can start performing coral IVF. So mixing the eggs and sperm so that they can fertilize and become little embryos. So those embryos start to develop. You can see them start to spin because they're developing little cilia. And then within just a few days, about 2 days, they become little swimming larvae. And then a few days more, maybe like 4 to 7 days, is when those larvae start swimming around and looking for a place to settle down and start growing as that first primary polyp. So once we have little polyps, we introduce little baby urchins, which are also spawned at Cal Academy, and the urchins keep the polyps nice and clean. They eat the algae. And then we introduce the algae we want, which are the actual symbionts. So what you just saw was a spawn that occurred in 2019, and you saw those first little primary polyps. And now the video that I'm about to play for you is 2 years later, and those little primary polyps have in fact become juvenile coral colonies. So this video was taken in June of 2021. So these colonies at that point were 2 years old. They're now almost 4 years old. They're even bigger than this. Some of them are still in the Coral Regeneration Lab, but some of them are actually out in the Philippine Coral Reef tank on public display at Cal Academy. And because they're now almost 4 years old and some of them are getting to be pretty big, we are hoping that eventually, maybe within the next year, maybe 2 years, these guys will in fact themselves start to spawn. So this leads to kind of one of our really exciting recent developments. In November of 2022, so just 2 months ago, we published kind of the first paper out of the Coral Regeneration Lab, in which we sequenced the parents and the offspring from 2019 and 2020. So what I mean by that is we took the genetic material of all of the parents that spawned in 2019 and 2020, as well as all of the babies that survived to be at least 1 or 2 years old, because they were sampled in 2021. And so with that information, we were able to create family trees. So we now know how all of these little babies are related to each other. Guys, I should really stop calling them babies. They're almost 4 years old. These are like toddler corals at this point. And we were able to start learning about how the toddler corals born in the lab might be different or similar to their, wild-born parents. So the first thing that we learned from the 2019 corals, of the 23 babies whose genomes we sequenced, 22 out of 23 are full siblings, so they had the same 2 parents. So this indicates to us immediately that 2 of these parents were way more successful in producing surviving offspring than the other 2. So that starts to raise questions. Why were those 2 parents more successful? Is it because they just produced more eggs and sperm? Or is it because they had some kind of genetic predisposition that made their offspring more likely to survive in our aquarium system? We found a similar thing in our 2020 cohort. So the first thing we noticed is that there was a coral love triangle happening in 2020. There are a couple of full siblings from these 2 parents, but then some half siblings from these pairings as well. But in this case, there were multiple colonies, multiple parents that spawned but produced no surviving offspring. And so what we have really started to investigate is why that might be and in what ways the offspring born in the aquarium are different from their parents. And one thing that we've really identified is that the way in which they're different actually appear to be in genes related to symbiosis. As we've already talked about, symbiosis with the algal symbiont is incredibly important for corals. So this is really what our next step is going to be, is trying to figure out how and why symbiosis in the aquarium is different from in the wild. And that has real implications, right? Because if we're going to start trying to breed corals in aquariums and then outplanting them to the wild, we need to know that they're going to be able to survive and that they're not so different that they're going to die as soon as you put them back in the wild. The other really exciting piece of news that just happened last week is we had a second paper come out in the Proceedings of Royal Society B. And for this paper, first of all, we assembled an entirely new genome assembly. So we created basically like a genomic resource for coral scientists around the world to be able to use to study genetics of their corals. And we also developed a new hypothesis. And I can't get into all of the details right now, but basically what we believe that we have found support for is that corals have stem cells that are very different from like human stem cells, in that we think that they can regenerate themselves and keep differentiating into different cell types throughout the entire lifetime of the coral. And this has really interesting implications for how corals might actually be able to adapt pretty quickly and like evolve faster than a single generation, which is kind of mind-bending and kind of very different from how we think of evolutionary biology. But it leads to really intriguing new directions for how we can think about adaptation of corals in this rapidly changing world. So those are two really big outcomes of our first two spawns of Acropora hyacinthus corals from Palau. And now I want to shift into the most recent spawns, so 2021 and 2022, of Acropora millipora from Australia. So in October of 2021, so a little bit over a year ago now, we started import corals of a different species. And these came from the northern Great Barrier Reef in Australia. So they were shipped to us. You can see my colleague Shale and I unboxing our corals. You can see my colleague Lisa checking them out as they just arrived, and Shale tagging them and taking genetic samples right as they arrived. So this is what they look like when they were set up in the lab. And this is actually what it looks like. So these are two of our amazing biologists, Lisa Larkin and Ryan Schaefer, setting up the larval rearing cones. The other piece of the setup here is not as pretty, but it is a conference room that we have transformed for the last 2 years now into our kind of mad scientist laboratory where we do a bunch of experiments. So in October of 2021, we had our first successful spawn of this new species from Australia. And we got this really beautiful video of it. So here is our coral releasing its gametes in our aquarium. And I will emphasize again, this is not a slowed down video. This is actually what it looks like in real time. And this is actually what it looks like when we're not filming for it to look pretty, but when we're actually doing the science. So we actually put nets on our pretty corals and capture the gametes that way. So that way we don't actually have mixing of the gametes until we want them to. So basically the gametes kind of funnel up through the net into a funnel and into a little collecting tube, because these gametes are positively buoyant, so they keep floating up until they have nowhere left to go. So this is what it looks like with us. This is in 2021, doing a bunch of the fertilization. So this is really just a bucket of hundreds of thousands of eggs and tens of millions of sperm. And then this is what it looks like a couple of days later. So we actually have our little swimming larvae living in those larval cones that Lisa and Ryan set up for us. So you can see here we had so many embryos. This was an incredibly successful spawn for our first time doing spawning of this species, and we were very excited. So we did a couple of different experiments that year. One of them was a temperature experiment. So what we did was we had a set of coral larvae that we held at normal temperatures, so about 26.5 degrees Celsius, which is just under 80 degrees Fahrenheit. And then we had a set of corals that were exposed to much higher temperatures, so 34 degrees Celsius or about 93 degrees Fahrenheit. So they underwent this temperature ramp for about 30 hours, and then they were all brought back down to normal temperatures and exact same conditions after that. We introduced these settlement tiles. So these are tiles upon which the larvae actually like to settle. And then after several weeks of having the little coral recruits, we took photos every couple of weeks to see how successful our little coral babies were that had either been at normal temperatures or at high temperatures. One of these tiles, by the way, is 7/8 of an inch in diameter. So this tile is very small, which gives you a sense of how tiny each one of our little coral polyps is. But anyway, we took hundreds and hundreds of photos, and I have an incredibly dedicated intern, Ariana, who has been painstakingly going through every single one of our photos, tracing and calculating the surface area, so the size of each of our little coral babies at every time point on every single tile, and counting how many corals there were at each time point. And the thing that we found that's really intriguing is that while higher temperatures, yes, definitely affected coral mortality, that is, a lot fewer corals were able to initially settle when they were exposed to higher temperature, over the course of a couple of weeks, the mortality between the two treatments ended up being about the same, which means that the ones who survived that initial heat treatment didn't seem to have residual negative effects later, compared to the normal treatment. And so we think this is a really interesting indicator about how we may eventually start to experimentally evolve corals that that potentially are more adapted to surviving higher temperatures. So that's something really intriguing. One other thing that we did is we collaborated with Manu Prakash and Ian Ho, who are at Stanford, and they put our coral larvae on what's called a gravity machine. So it's this incredible microscope that they have developed, in which you can watch the coral moving in 3 dimensions. So unlike a normal microscope, the coral is totally free to swim wherever it wants. In this case, it's swimming down. But we're able to measure all sorts of interesting things about larval behavior and physics and fluid dynamics using this machine. So that's been a really fun project that we've been working on together. But now I want to talk about this year, or I guess rather now it's last year, but the last 2 months in the Coral Regeneration Lab. So in November of 2022, just about 2 months ago, the full moon was on November 8th. I should back up for a second. In 2022, for the first time, we did not import new corals into the Coral Regeneration Lab. The only corals we had were the ones that had already been imported in 2021. And so those are the ones that I was just showing you had their super successful spawn. And what we really wanted to test for this year, 2022, was whether we could keep all of those animals happy and healthy and go through their entire gametogenic cycle within our system under our, very simulated artificial parameters, the lighting, the temperature, all of that, and still have a really successful spawn. So this was the test. So in November of 22, the full moon was on November 8th. So we started watching and waiting around November 13th or so. And we expected to probably see coral spawning around day 13 after the full moon. So that would be day 7, 8, 9, 10, 11, 12, 13 was kind of what we were estimating. And sure enough, we had coral spawning happen on day 14, 15, and 16. So only 2 corals went those 3 days, and it was actually the same 2 corals, which is interesting. The other thing that's really intriguing about this is that those 2 corals are quite genetically different, because we've now sequenced the genome of all of our animals, than everyone else in the system. So potentially they're actually a different species. But anyway, in November of 2022, only 2 of our corals spawned. The 2 that are quite different, potentially genetically, from the other ones. But then we were like, okay, wait, but what about our other 9 corals? Because we had 11 of them in total. And we started chatting with some of our other colleagues who were studying the species, both in Australia and in aquariums in England. And they also said, yeah, only a few of our corals went this year, so maybe they're waiting for the full moon in December instead. So we started looking at the December calendar. We saw that the full moon was again on the 8th. which means we were starting to get worried that our spawning was going to happen maybe the 22nd, 23rd, 24th, 25th, like really hitting up against that holiday weekend, which we weren't thrilled about. So Rebecca and I made a decision to mess with the moon a little bit in our system. So what we did is created a simulated full moon. So instead of having the lights simulate the full moon on December 8th, we told them to go early, make the full moon happen on December 5th instead, with the hope that we could get the corals to spawn a couple of days early. We had no idea if this would work or not, but lo and behold, it sure did. So 7, 8, 9, 10, 11, 12 days after our fake full moon, our corals spawned on the 17th, 18th, 19th, and 20th. So this was very exciting because it really does indicate the amount of mechanistic control we have in our system to influence the spawning of these corals. So we had 2 corals go the first day, then 3, then 6, then 4. We're thrilled with these results. And let me show you a little bit of what that looked like. So once again, beautiful coral spawning. Once again, this is in fact in real time. And the thing that we were really interested in doing this year was to do some preliminary testing of interventions to increase growth and survival of really early juvenile corals, so within the first few weeks of life. The reason for that is that in the beginning of life, both in the wild and in the lab, less than 10% of coral larvae will actually successfully settle, like successfully metamorphose and find a place to settle. And less than 10% of those corals that initially settle will actually survive to their first birthday. So the bottlenecks here are huge when we're thinking about trying to scale up restoration interventions in this way. And so the key thing that we're trying to tackle is to increase growth and survivorship of baby corals at these really critical bottleneck steps. So the first way we're trying is by seawater buffering. So that is to increase the alkalinity of the seawater. The reason for that is that when corals undergo their initial calcification stage, so making that calcium carbonate skeleton, it's really energetically taxing. And so people think that a lot of the reason why there's a lot of death at that stage is because they've used all of their energy reserves on that. So by buffering the seawater, we thought, well, maybe it'll make them use a little bit less energy than they would otherwise. So that's the first thing we tested. The second thing we tested was to try to feed the larvae with amino acids. So providing them food, even though usually these larvae don't really feed during their first few days of life, we thought, well, maybe if we can give them a little bit of nutritional advantage, that might help them. So that was the second intervention. And then the third one was to try to get the little larvae to uptake algal symbionts earlier than they would usually. The idea being maybe the symbiont can give them more energy and get them through those energetic slumps in their first few weeks of life. So those are the 3 things that we're currently testing. We are, once again, measuring hundreds of little baby corals, including today. That's what we were doing, taking photos, measuring their growth, measuring their survival, to see which of these interventions might actually really be successful. And this is from this year, so this is just a few weeks ago, all of these tiles back in the lab, lots of little baby corals next to their baby urchins on their tiles. But now I want to take us from the lab to the ocean. So hopefully you're excited. You're like, wow, Elora, you're doing lots of cool work in the lab. But maybe, hopefully, you're thinking, okay, but this is all happening in the lab. How do we translate this to the ocean? And the way that we started by doing that is partnering with Roatan Marine Park. So Roatan is an island off the coast of Honduras. And Roatan Marine Park is a nonprofit organization that manages the coastline and the resources, marine resources of Roatan. They've experienced a lot of disease. Really within the last 2 years, they've lost a ton of their corals to stony coral tissue loss disease. But they've also done an amazing job of documenting the disease, trying to come up with treatments for the disease, and doing their best to protect their corals in their marine park. And they reached out to us in 2022 because they were planning a workshop in September of 2022 to train folks in Honduras, so from the government, from other nonprofits, all across the country, to learn how to do assisted sexual reproduction of corals in Honduras. So this was the first ever coral sexual restoration project to ever happen in the country. So this is our group. So me and my colleague Lisa, as well as all of our colleagues in Honduras, as well as from CoRL, which is another group that's doing a lot of coral reproduction work in the Caribbean. And we created these what are called cribs. So they look like giant trampolines, but really what they are these basins that can sit in the ocean, and we can put the little coral larvae into those basins so they can actually swim around and be in regular ocean water without actually, escaping into the ocean. So we filtered a bunch of seawater, got everything ready to go. We made a bunch of nets similar to the ones that you saw in our lab, but much larger because they were going on actual coral colonies in the wild. And then we got ready to do a bunch of coral spawning dives. So our colleagues in Roatan had already figured out that the corals that we were interested in spawn at around 9 p.m. And they expected it to happen within, a pretty short window after the full moon in September. So we would dive down to about 20, 25 feet of depth, and we would sit at our designated coral colonies. We would then, if we saw them about to start spawning, put a net on them, capture their gametes, and then we would end the dive by bringing those gametes to the surface. So this is an amazing video that my colleague Lisa took in the wild. So this is her on scuba watching this incredible colony. So you can see it's very different from what our other species in the lab does. Rather than that little trickle of gametes, they all get released at once. I can't even tell you the amount of patience that Lisa had to have waiting for 20 minutes underwater, holding her camera, getting ready for that one moment. But it was pretty spectacular to see it. So we were thrilled. We saw coral spawning, and the first day of spawning was on Honduran Independence Day. So we were very excited. This is our team having just gotten out of the water and onto the boat with our coral gametes in tow. And then we did a lot of exactly what I just described to you in the Coral Regeneration Lab. So we fertilized the coral gametes. We checked for embryogenesis, so cell division and development on a microscope. This is Lisa and I. I will also add, by this point, it was about 3 a.m. Coral spawning is late night work. So this was us with our 4-hour-old embryos that we were preparing to then bring back into the ocean. And this is what it looks like. So this is the inside of that crib. You can't see the little corals because they're way too tiny, but they were swimming around in their basin. There's a bunch of settlement tiles for them to settle on down here. So this was a really successful first, coral restoration via sexual reproduction event in Honduras. And we're really hoping to foster and generate more collaboration with our friends down in Roatan to continue this work. All right, so I'm going to wrap up here just by saying, what are we learning / what will we learn from the Coral Regeneration Lab? There's a lot of things. So first, we've assembled a reference genome from coral sperm, which is a great resource for coral geneticists around the world. We've learned how mutations are inherited from parent to offspring. We've learned which parent genotypes are most successful in producing surviving offspring. We're starting to understand how the lab environment might select for different traits than the wild. Oh, I didn't really talk about this one, but we have a partnership with NASA where we're learning about how gene expression changes during larval metamorphosis. We're starting to learn how, where, and why larvae swim and settle, how temperature affects larval settlement, which interventions might increase growth and survival rates at really critical life stages. And then finally, and really most importantly, how we translate all of these findings into regenerating reefs at scale within the ocean. So with that, thank you so much for joining me. I have so many collaborators and wonderful people to thank on this slide and beyond it. But for now, I guess I'll stop sharing my screen and be happy to take any questions you may have.
Awesome. Thank you so much, Elora. Gosh, I always learn so much from you. And it's nice to see our little toddler corals. We do have a couple of questions. One, I think, just a clarifying question. That last video you shared, Amy was curious, was that at night?
Yeah, so that was taken at night around 9 p.m. So like maybe 3 hours about after sunset. Yeah, so those were all night dives. So we're going down there holding 2 flashlights plus our cameras and all of our other gear. Yeah, it's totally dark without our flashlights.
Wow, quite the setup. I bet it's so amazing. Another question from Amy. So they shared that they dive a lot in Roatan. Is there any opportunities for volunteers?
Oh, I bet Roatan Marine Park would be very happy to take you as a volunteer if you email them. I believe they actually even have a certification program, so you can take like a certificate course with them and like become like a coral restoration certified person.
Wow, that is awesome.
Yeah, you should definitely reach out to them.
And that was the Roatan Marine Park?
Yep. Mm-hmm.
Okay, very cool. Let's see, a question just came from Elizabeth. In Honduras, what was the advantage of fertilizing above the surface versus letting it occur unassisted underwater?
This is a great question. So unfortunately, the reason that this all is kind of necessary at this point is because so many corals have died in Roatan. So as we mentioned earlier, the reason that corals all, reproduce at the exact same time is that you need to be able to find a partner, right, in the very large ocean. And when the corals are at way lower densities, so many of them have died that there's way fewer corals next to each other, it really diminishes the chances of a single sperm finding a single egg from 2 different colonies in, the vast ocean. And so the reason to do the collection is for us to increase the chances of successful fertilization. So we collect the gametes and put them in a small bucket so that they, have way higher chance of finding a mate. So yeah, I mean, in a world 50 years ago, this totally was not necessary because of the density of corals on the reef. But as that density decreases, it becomes more necessary.
Wow. So interesting. I think that was the last of our questions. Members, if you have any other questions, please feel free to drop them in the chat. I did have one question that came up. You said that some of the corals that we have spawned in the lab were actually in the Philippine Coral Reef at the Academy. Are there any identifiers or ways that we could find those?
This is a great question, Jess. We should work on making that happen. The answer is right now no. And they're very small. I'll be honest with you. Even I have a hard time seeing them.
Okay.
When I'm looking at them, like I know for sure where one of them is, but some of the others, like I have a hard time. I'm seeing it, but okay. I promise they're there.
Love it. Maybe in the future we'll be able to see them.
Yeah.
That's fun.
As they get bigger, they'll be easier to see, I guess.
I love it. Let's see. Wendy just popped in a question. Can you talk a little bit more about how the coral stem cells can allow for evolution within a coral's lifetime and how that might affect adaptation to warming?
Yeah. Oh man, Wendy, I don't have enough time. The short answer is that in some ways, there's a lot of parallels actually to thinking about human cancers. So evolutionary biologists have, within just the last few years, been really interested in studying cancer evolution because, similar to a coral colony, the way that a cancer tumor in your body evolves is that, a cell accumulates a mutation and then it starts dividing. So all of those cells have those mutations. And there's, selection on those cells themselves. I don't know if I'm explaining this well, but a similar thing happens we believe, in a coral. So polyps that accumulate mutations that are either neutral or beneficial will survive to reproduce, whereas polyps that accumulate detrimental mutations will not, right? And so you can imagine that if you get a bad mutation, that mutation actually shouldn't proliferate through the colony because the polyp just dies. So another way to think about this is when we see coral bleaching, oftentimes what you see on the reef is the only part of the coral bleaches and the rest of it survives. And so what we think may be happening here is that there are mutations accumulating within a single coral colony, and that when something like a bleaching event comes through, then that actually is a selection event that's happening not on the level of the colony, but on the level of the polyp. And so it's the polyps that survive, that have those mutations. And because of this interesting stem cell thing that I can't fully get into right now, those mutations are actually passed on to their offspring. It basically makes the adaptation process happen a little bit faster than it would otherwise, because basically there's a little bit of pre-adaptation happening within the colony before those mutations even get to the offspring.
Wow. Sounds like we could do a full other talk.
I would be happy to.
Okay, we'll talk about that. Let's see, we've got like 3 more questions. Do you have time, Elora?
Yeah, sure.
Awesome. Let's see. Have you heard about the surprising number of corals in the Tala Bay, which is near Roatan? It looks like they shared that this is the last place you would expect to find corals because it is so polluted with runoff from untreated sewage and agriculture. Have you heard of this bay?
What was the name of the bay?
Tala Bay. Tala?
Oh, Tela, yes.
Tela.
Yes, actually, there was someone from Tela at the workshop, and he was very excited to tell us all about how great their reefs are. Yeah, I feel like we're basically doing a Honduras tourism exercise right now. Everyone should go check it out. But yeah, no, it is an interesting story. And I think that they definitely want researchers to start coming to Tela and trying to figure out how and why their corals are surviving there. So yeah, I'd be happy to check it out.
Interesting. Let's see, from Mike, can dead coral be replanted?
So a dead coral is no longer alive. And so while the skeleton still exists, there's really no, like, you can move a skeleton around, but that's not going to make it become alive again. But I think some people have done some work to try to see if corals like settling on the skeletons of dead corals. So, like, will a larva settle on dead coral? I think the answer is often yes. So there are potentially interesting ways to think about what you can do with a dead reef. But yeah, you can't, unfortunately, make it come alive again.
Very interesting. Let's see, last question from Amy. How has the cruise industry changed corals? She says she's seen a ton of bleaching in Roatan since the big boats have come to the island. Do you know more about that?
I don't know. I mean, certainly we know that with a cruise ship comes all kinds of pollutants, which, affect water quality and can affect coral health in lots of negative ways. I don't know if there's been studies done linking bleaching to cruise ships, but that would be a really interesting thing to follow up on. So I'm not sure.
Very cool. Well, I think that is the last of our questions. Thank you so much, Elora. Sounds like we might have some follow-up member talks. Lots of thank yous coming in. I think folks really appreciated learning more and hopefully maybe coming to the Philippine Coral Reef at the Academy and checking out some of those toddlers. But thank you so much for joining us, and thank you, members. We'll see you next time.
Great. Thank you.
Bye.
See ya.